All right. Now, I'm assuming that you've had enough chocolate and coffee to be awake for the afternoon. And if you haven't, this gentleman will wake you up because he actually is relatively interesting. Now, I know him well enough to say relatively. His name's Dr. David Cribbs with UCI Mind. He's a professor in residence, neurology at the UCI School of Medicine. He's associate director of UCI Mind. By the way, he's really good with red wines, if you have any questions. Actually, I'm not kidding. He really is. He receives his Ph.D. from the West Virginia University. His research interests include Alzheimer's disease, immunotherapy, cerebral vascular pathology, and neurodegeneration. Currently involved in the design and development of beta-amyloid immunotherapy as an experimental approach to test the amyloid cascade hypothesis of Alzheimer's disease and as a potential intervention for the treatment of Alzheimer's disease. I'm not going to read the rest of his bio. Suffice it to say, he is an incredibly accomplished scientist. I only am not reading it so we have more time to hear him talk about his work. Please join me in welcoming.
Thank you, Jim, for that kind introduction. I'm not going to thank the organizers of the conference. What I'd like to do is thank all of you for staying to hear the last talk of the day. And I don't have any Star Wars cartoons or anything. I feel inadequate up here. But I am going to talk about, I'd like you to focus on three things here that have some importance. And one is the title. The other one is a picture here, and that's a schematic or a diagram of a DNA molecule. And what I'm going to be talking about, if you go up here to immunotherapy, I should have substituted the title, in the title, the word vaccine, because that's what I'm going to be talking about, the concept of a vaccine for Alzheimer's disease. And you've heard a lot of talks earlier in the day, and I'd like you to go back and remember, I'm going to pull a Rick Perry here, three things about the talks earlier in the day. Hopefully I can get all three of them. Rick Perry, I think, got, what, 2 out of 3 or something? Anyway, the first one is that one of the first lectures today was by Adam Flesher, and he was talking about when they proposed that the amyloid occurred first, and then the tau, and all these other events occur in the brain, and that the clinical picture, the clinical development of the disease starts very late, such that the amyloid accumulates 1 to 2 decades before you begin to see clinical features of the disease. And I'm going to have an example of similar to some of the slides that he showed earlier in the morning to perhaps convince you the reason that immunotherapy so far has failed in the clinical trials, and I think probably some other drugs have failed as well, simply because they were started too late in the disease process. And you heard Charlie Glabe talk just before me. He was talking about, if his model is right and these neurons are dropping out and that's why you're seeing these plaques, then those cells are gone. Those processes are gone and those synapses are gone. So you're asking a lot of a therapy to go in and start treating people in their 70s and 80s to reverse this whole process. So I'm saying that up front because my research, one area of my research is this process of this idea of immunotherapy. And if the clinical trials have failed, there's been some positive indications, but it certainly hasn't been convincing. So I'd like to argue that the therapies have been tried too late, and perhaps the antibodies were targeting the wrong part of the amyloid peptide, or maybe the wrong conformation of the peptide, referring back to Charlie Glabe's talk. So I've already mentioned the two things here, and the other thing is that's my name, but you can see how long my title is. And what that tells you, like going back to Charlie's presentation, how many arrows you have is how confused you are. The more things you have under your name, that's an indication of how long you've been someplace. So in August, it was my 20th year as a faculty member here, and it's actually 21 years. I came out here on sabbatical 21 years ago for 8 months. And I like the weather, I like the palm trees, and Carl Kottman was gracious enough to offer me a position in his lab. So that's my way of introduction, and so what I'd like to do is actually move on. These are my disclosures. I do some consulting, and like Claudia Kawas, nobody's paying me right now, although I get, I think, $400 for every meeting of the DIAN, that's that dominantly inherited Alzheimer's network, to try and select drugs to go forward in clinical trials in individuals that have these dominant Alzheimer's mutations. And there's no content here that was industry produced in my presentation. Okay, so Marcelo, about a week ago, told me I had to have some course objectives, and so That's what these are. And most of them are pretty evident. You've been here all day, right? So identify Alzheimer's disease therapeutic targets. Okay. I've already told you I'm going to try and develop a vaccine for Alzheimer's disease, and my target in this presentation is going to be A-beta. I should tell you my lab and other labs around the world are targeting things like tau pathology, alpha-synuclein, and so those efforts are ongoing. And whether they're going to be effective, whether they're going to help improve people, or whether they're going to make people worse is still an open question. One of the amazing things about transgenic mouse models of Alzheimer's disease is we've cured them. I don't know, I'm looking at Andrea Tenner, maybe 100 different therapies have been effective in mice, but we haven't had that success in humans. So hopefully that will improve as we learn more about the disease. I'm going to present a very brief introduction of how immunotherapy might work. I'm going to discuss how immunotherapy we think reduces amyloid plaques in the brain. I'm going to very briefly introduce the concept of active versus passive immunotherapy, and there have been both active and passive immunotherapy clinical trials in humans, okay? I'm going to explain the difference, I hope, between protein and DNA vaccines, because a lot of my work is focused on DNA vaccines, and so I need to explain to you what a DNA vaccine and how it works. I'm going to provide a summary of the human immunotherapy clinical trials, and I'd like to discuss the potential advantages of early intervention for immunotherapy in Alzheimer's disease, and perhaps I've already done that in my introduction. Okay, so this was an overview, which is essentially the same thing as what I just told you, the course objectives. But there's a few other things here. I'm going to talk a little bit about the pros and cons from immunotherapy clinical trials, and I'm going to try and this is going to be a little bit difficult, but I'm going to try and provide a rationale for what we call an epitope vaccine. And you need to remember, I already mentioned one of those things in my Rick Perry moment, the imaging from Adam Fleischer. I'd like you to remember Andrea Tenner's talk and also Matthew Blurton-Jones's talk. Andrea introduced the concept of adaptive immunity. Okay, there's innate immunity, there's adaptive immunity. And in adaptive immunity, we typically talk about B-cell and T-cell responses. And in the immunotherapy, I'm going to rely on the production of antibodies, okay? And you've heard about these antibodies. You've heard about antibodies being able to recognize pathology by immunostaining. And I think let me finish this up real quickly. I'm not going to talk about memory T cells. I don't have enough time today. This concept was based on the idea that going in to immunize elderly individuals where they may be affected by what's called immunosenescence, you've all heard that flu vaccines don't work as well in the elderly. So the idea was to take advantage of existing memory, T-cell memory, to enhance the vaccine response in these individuals. But I don't have time to deal with the data that we have on that. I'm going to talk about experiments we've done at mice, rabbits, dogs, and non-human primates today. And so we're looking to move forward in a clinical trial. We'd like to be confident that we might have a therapeutic benefit, and the thing that's always in the back of my mind is I hope that we don't make people worse. And you've heard from Charlie Glabe today that you're not always successful and things don't always turn out as you hope. Okay, and I'll briefly, if I have time, talk about the clinical trials that have occurred already and have been largely disappointing results. So let me go back and my brief introduction to immunotherapy. What I'd like to do is introduce the concept of a snake bite. My wife's in the back of the room, and I thought about talking about zombies because I was interviewed a week ago trying to explain how I would develop a vaccine against zombies. But I think if I stick to the snake bite, I'll have better success. So here goes. So all of us are familiar, if you get bit by a snake, like a rattlesnake, a snake that has a toxin, that if you get to medical help and if that place has an antivenom and they give it to you early enough, they may save you from death or from disfigurement or disability or something like that. And this antivenom has antibodies. And So we need to keep in our minds the concept of an antibody molecule. And we're not going to have time to describe all the wonderful features of an antibody, but it has the ability to bind to things, and it can recognize the chemical structure. And anti-venom antibodies, these antibodies recognize the toxin, and they neutralize the toxin. So the idea with immunotherapy for Alzheimer's disease is have antibodies get into the brain, bind to this peptide, this A-beta peptide, and neutralize the toxic or aberrant effects of this peptide. And ultimately, there's good evidence in both mouse models, canine models, and humans that you can do this. Ultimately, the bottom line here is going to be whether we can block the onset and progression of the disease, and that's still an open question. Okay, so we've already seen this. That would be the test, which one's the healthy and which one's it's labeled, so it's not really much of a test. You've already seen all this stuff, so I'm going to skip over that. These are senile plaque, and Charlie already mentioned the fact that it not only accumulates in the parenchyma of the brain itself, but it also accumulates in the vessels and disrupts vessel function. I'm not going to go over this because Charlie did a good job, although he skipped some things like trisomy 21 and Down syndrome. They have an extra copy of that chromosome. They have an extra copy of the amyloid precursor protein, and they develop all of the pathological features of Alzheimer's patients. Okay, and there's other reasons to believe that this amyloid precursor protein is involved in the disease, and because of many of the mutations surround or in the A-beta peptide, I think there's really good evidence to support the idea that this A-beta peptide is causative in some fashion. I'm going to skip the rest of this. And so the general concept was these anti-A-beta therapeutic strategies, and Charlie talked about some of them. You either want to block the assembly or to block the aggregation, you want to enhance clearance, or you want to reduce production of this peptide. And I'm going to skip all these, and I'm going to focus on immunotherapy, because that's what about 50, 60% of my lab's effort is on immunotherapy. So that's what I'm going to focus on. And to further introduce the concept of immunotherapy, I'm going to introduce a canine model. And this collaboration was done with Elizabeth Head, who's now a faculty member at the University of Kentucky. And really what you need to know here is that elderly dogs develop amyloid deposits like humans. Now there's differences. They don't develop the tau pathology, and the amyloid deposits are diffuse by nature. So there's differences. But what this is showing is a clinical trial that we did in canine animals. So these were beagle dogs. They were 8 to 12 years of age, and we followed them for essentially 2 years, giving them immunotherapy, just like was done in transgenic animals, also done in humans. And I'm not going to talk about all the arms of this study, but what I would like to show you are two things. So these are brain sections from 12 to 14-year-old beagle dogs showing extensive amyloid deposition. And this is a representative slide from an animal that was immunized for 2 years with this vaccine. It was just the amyloid peptide plus alum, a conventionally available adjuvant. And this is just quantification of the blue shows the amyloid levels in these various brain regions, and the red shows those animals that received the vaccine. And this was the reference, and published in 2008 in Journal of Neuroscience. And I might add that we never saw any adverse events with this vaccine design. But unfortunately, and just briefly, I don't want to go into too much detail, but this is what a schematic of an antibody molecule, a heavy and light chain. And here's the A-beta binding to the antigen binding site. And so there's lots of research in my laboratory and many other laboratories looking at how these antibodies get into the brain, how they may interact, bind to the monomeric, fibrillar, and oligomeric forms of A-beta, how they might decrease intracellular amyloid that Charlie Glade was talking about. So there's a bunch of different mechanisms here: peripheral sync, microglial-mediated clearance, and this is still under active investigation, but ultimately it really doesn't mean much if we don't get a clinical benefit with immunotherapy. This was particularly depressing to me when the phase 2 clinical trial by Elan, the first immunotherapy trial, was an active immunization. So they used a full-length A-beta peptide, and they used a very strong adjuvant. And the immunized humans, phase 1 was safe. There weren't any issues. In the phase 2, they decided to reformulate the vaccine, and the result was this. Some people thought the idea was bad, a bad concept from the very beginning, and you can see their title of this letter to the editor, Ill-Fated Amyloid Beta Vaccine. And the adverse events that halted this clinical trial were very serious. About 6% of the individuals developed subacute meningoencephalitis. And so at this time, my colleague and collaborator, Michael Agajani, and I had submitted a grant to NIH, an R01 application with all these great ideas of how to make a better vaccine. This came out, and you can imagine what the critiques were like. This failed, why bother? So that was very disappointing to us, and we wrote a rebuttal and ultimately got the grant funded. So we were successful in that. And the general thinking is the reason that this vaccine failed was there was an adverse T-cell response. Remember that adaptive immune response that Andrea Tenner was talking about? So to get a good antibody response, you not only need B cells to produce these, I'm skipping a couple of steps here. Charlie likes to keep the arrows down to a few, so we can make you believe that we actually know what we're talking about. So there's multiple steps here. The B cell, there's plasma cells, and they are factories to produce antibody molecules. But that's where we think is the important part here. Antibodies that bind to the A-beta peptide and are capable of reducing the pathology in the brain. What we need a T-cell response to get a strong antibody response. And if you can keep that in the back of your mind, that's going to be important when I talk about that concept of that epitope vaccine, okay? Here we think, and it's not been proven, that what happened was you got a B-cell response and you got a T-cell response, and now you got a situation where you have an autoimmune response, similar to MS, multiple sclerosis, where you have T-cells actually attacking your body. So it's your immune system attacking your own body, and like I said, that hasn't been proven. It's interesting that Elan didn't have any problems until they actually reformulated the vaccine and included polysorbate 80 here. And one injection in an individual that had received 4 immunizations of the original formulation, no adverse events. Reformulated vaccine, 1 injection, and the patient developed this meningoencephalitis. So, and I've already told you that we had submitted this grant application and it was bounced back and said, why bother? And so we sat around and thought of potential problems with the immunotherapy that Elan had come up with. And then we tried to come up with potential solutions so we could go back in and get funding and continue the research. And I'm not going to focus on all of these. I mentioned in a previous slide there may have been a T-cell mediated autoimmune response. And so our thinking was to eliminate the T-cell epitope in the A-beta peptide. So remember, they used the full-length A-beta peptide, okay? And what we're going to do is we're going to cut the A-beta peptide into little pieces and find a piece that's good for producing antibodies, and we're going to try and identify the pieces of the A-beta peptide that are responsible for producing the T-cell response. And then what we're going to do is we think we're pretty clever. We're going to substitute in other T-cell epitopes to drive T-cell response to help produce the antibody production, okay? And so the idea is that the T-cells won't recognize the native A-beta or the native APP molecule that the A-beta came from. And that, I hope I did a good job explaining that, because that's sort of critical to what's going to come after this. Okay. And so we're going to try and make a better vaccine by getting rid of the T-cell epitope in the A-beta peptide. And the way we did this a number of years ago was, and you've already seen this is the amino acid sequence of this 42 amino acid peptide. And what we did was we synthesized a lot of shorter peptides that were overlapping. So we're going to go into the laboratory and we're going to measure what the antibodies are binding to, and then we're going to stimulate T cells from the animals that were immunized, and we're going to find out what the T cells recognize. And what we're going to do is we're going to cut this peptide up, and we're going to keep the portion that the antibodies recognize, and we're going to use that as our B cell epitope, and we're going to eliminate all the other sequences here that might induce a T-cell response, okay? So, all right. So this is very old. So what we found was in mice, and I don't need to go into all of this, this was just different adjuvants that we used to try and induce a strong immune response. We found that the B-cell epitope was in the N-terminus region, this A-beta 1 to 15, and in this particular mouse strain, the T-cell epitope was very close. Actually, most of the data on humans suggests that the T cell epitope is further down in the peptide. And so our idea was then to develop an AD epitope vaccine, and we used peptide. These were our early stage developments with the vaccine concept. We used multiple antigen peptides, and we also started working with DNA vaccine technology. And we're just using a circular piece of DNA, and then we insert a sequence of DNA that contains the vaccine. And so the fundamental difference that we should appreciate is with a peptide protein vaccine, you're delivering the vaccine. With a DNA vaccine, you're delivering the information that contains the vaccine. So we're going to ask the animal or the humans that will receive this DNA vaccine to take this DNA up in their cells and actually produce the vaccine themselves. Okay, so that's the fundamental difference. The DNA is taken up by the cells if you do a good job, and then they'll express the protein, and the B cell epitope and the T cell epitopes, and you hope that this is going to induce a strong antibody response, and I'm going to show you, hopefully, examples of that. And these are just some papers that we published years ago looking at these issues. This was a T cell epitope that I found in the literature. It always, as Matthew Burton-Jones mentions, it's good to read the literature. You can learn things and incorporate them into your own research. And so this was a synthetic promiscuous T cell epitope. And so synthetic is it didn't occur in nature until these people in San Diego developed it. And what was interesting about it being promiscuous is you'd like to have a vaccine that everyone in the human population would respond to rather than 5% or 10%. You don't want to have to make 50 or 100 vaccines and then identify who's going to get the vaccine. So if you can develop a vaccine that has multiple T cell epitopes, you're going to be confident that in the vast population of humans that the vast majority of them are going to produce an immune response to your vaccine. Okay, and we're going to define that B cell epitope, that the sequence of the peptide that we want to produce antibodies, the therapeutic antibodies. So that's the concept. And this was Padre, and I mentioned it was just a synthetic epitope. We've continued to incorporate that in our vaccine. We've seen beautiful responses in mice, in rabbits and dogs, and in non-human primates and monkeys. Okay? So, and these are just some of the advantages of a DNA vaccine. It's easy to manipulate, it's stable, it's relatively inexpensive, except I think we spent $400,000 to make 600 milligrams of it, so it's not that cheap. It wasn't my money, but oops. And it can be easily engineered to incorporate what we call molecular adjuvants, and I'm not going to talk about that today, but the concept in a vaccine is you need a good immunogen, which consists of a B cell and a T cell epitope, and typically you need an adjuvant. And getting adjuvants approved has been difficult. Because things that typically induce a good immune response, as Andrea Tenner also explained this morning, they typically produce undesirable side effects, swelling and pain and those types of things, or you end up with lesions in your arm and granulomas and stuff like that, and they're considered particularly unattractive. So the issue of adjuvants is a problem. Okay, so, and this was our very first DNA vaccine, and it was so bad and produced no immune response, I was ready to abandon the whole project. We just took the A-beta peptide and put it into a plasmid with a promoter, injected it into mice, and we got nothing. So, but we kept working, and we incorporated a Helios gene gun. It's made by Biorad. And what this helped us to do was to deliver the DNA, our DNA vaccine, directly into cells. And so what this is a gun. And it has you have a helium tank, and you have a regulator, and you coat the plasmid, the DNA vaccine, onto gold particles, and you coat the inside of little plastic tubes, and you make bullets. And there's 12, you can load 12 bullets into this gene gun, and then you can blast these gold particles through the skin into the cells, and those cells begin to express the vaccine. Now, it's expensive, the gold particles are expensive, and it doesn't scale up to humans. And so what we did over time was transition to what's called electroporation system with a company in San Diego, ICOR, and this is their system, and it's a fantastic system. And what it does is it takes the DNA solution, and there's a syringe, and it injects it, and there's a series of electrodes, and it delivers a series of pulses that open holes in the cells. The DNA is taken up, and what you can see here are two different examples. is this fluorescent green, like Matthew Burton-Jones was talking about. This is the protein that was coded by the vaccine that went in, and all this is doing is a comparison of the gene gun, the one that looked like a real gun, and the electroporation showing that they both work. I'll pretend that there's a laser here. What I'm going to try and do in a couple of minutes is try and show you how our thinking about the vaccine design and construct changed over time, and how we made incremental improvements in the vaccine such that we could move into non-human primates and get what we think is a therapeutic dose of antibody produced with a DNA vaccine. Okay? So originally our our concept vaccine and our design had one copy of that B cell epitope, the N-terminal sequence of the A beta that we want to make antibodies to. And so we got low levels of antibody here. And I came up with this idea to increase the number of the B cell epitopes in the design. So you don't have to worry about all the stuff here, but just see the 3 there. So by including 3 copies of this, now we're getting much higher titers of antibodies. So so we're improving the design of the vaccine, the B cell epitope, by incorporating 3 or more of those into the vaccine design. And that's just shown up here as a diagram. So 1 A beta, or 3 copies of this B cell epitope. And this padre, remember, is that promiscuous T cell epitope. And so, yes, okay. And I'm going to skip a whole bunch of research. And what we did, and we got rid of this. This was our molecular adjuvant that we found out didn't work in rabbits, and it was going to be a problem to get it approved by the FDA anyway. So we eliminated that, and we began incorporating more and more T cell epitopes with the idea of being able to immunize the entire human population and get a good immune response in everyone that we immunized, hopefully. And these T cell epitopes came from various toxins and various vaccines that are already commercially out there. And people in this room have memory T cells to these particular toxins, and we're not going to use the toxin. We're just going to use the T cell epitope. So there's nothing toxic about the vaccine, just a T cell epitope that's going to drive T cell proliferation to try and help promote B cell transition from B cells to plasma cells to produce a lot of antibody against the A beta peptide. So that's sort of the general concept. So this is, and I've skipped once again a whole lot of research and a whole lot of data, but this is just showing rabbits, so we can use our DNA vaccine and we can get a nice immune response in rabbits with electroporation of our DNA vaccine. And this just shows that there are different scales because we didn't have a purified monoclonal antibody to measure the concentration here in the monkeys. But we were able to get, both in rabbits and in non-human primates, we were able to get very nice immune responses. Good antibody responses. And this just shows further transition. So Anahit, a collaborator, came up with some additional T cell epitopes. And this is our final vaccine that we've now purified, and we have a GMP grade. And we're looking to move forward, get FDA clearance to go forward with the vaccine trial in humans. I am not trying to... and this just shows you how further vaccine design, by incorporating additional T cell epitopes, we're able to get better antibody response to the final vaccine design than we are with this sort of second phase design here. So there's 3 B cell epitopes to produce antibodies, and there's all these promiscuous T cell epitopes to try to drive a robust T cell response to the vaccine. And this is just a summary of what I've tried to tell you today about our efforts to design a DNA vaccine to produce therapeutic levels of antibodies that recognize A beta. And I've showed you in that canine model. I didn't go back and show you the studies that we did in APP transgenic animals, but we were able to do the same thing with a DNA vaccine that we were able to do with a peptide vaccine, and that is to reduce the A beta pathology. And I should point out that when we went in a triple transgenic animal where they had A beta and tau pathology, when we immunized early, we were able to reduce the A beta pathology as well as the tau pathology in that animal model. And so I've gone over this. You know, I've told you about electroporation. I told you about some of the properties of a DNA vaccine. I've tried to provide you with the rationale and the evolution and optimization of our DNA epitope vaccine. And I did mention briefly the concept of using existing memory in the human elderly population to try and drive a robust antibody response to the vaccine. And so this is, we're not going forward with this U44, we're applying for a U1 to take the DNA vaccine to clinical trials. And we've collaborated with Lundbeck, a Danish pharmaceutical company, and helped them design a protein epitope vaccine which incorporates similar concepts. So there's multiple copies of the A-beta B-cell epitope plus a couple of different T-cell epitopes, and they're supposed to be, I said it's 2013, now it's been pushed back to 2014. So hopefully in the future we'll see how this concept of an epitope vaccine will work in human clinical trials. And this just is an illustration of how the DNA vaccine, so these are the thioflavin positive plaques in that triple transgenic animal, and after 6 months of immunization you can see there's a significant reduction in those plaques in the mouse brain. How much time do I have? Huh? Okay. So maybe I'm a little ahead of the schedule. I said that in the beginning that I would talk about the clinical trials that have occurred already, and I already told you that things haven't gone wonderfully with these clinical trials. I'm going to now go back and introduce this concept of passive immunotherapy, because everything I've been talking about up until this point has been active immunization. We're taking either a peptide protein vaccine or a DNA vaccine, and we're immunizing, and we're expecting the animal or the human to produce antibodies against the vaccine. In passive immunotherapy, it's analogous to the snake venom, where you deliver the antibody passively. Nothing is produced by the animal or the individual that receives this. It's an expensive way to immunize people, because what you need is to develop an antibody, typically a mouse monoclonal antibody that provides evidence you can see in an animal model that you can clear the pathology in the brain. And then you have to go through steps where you humanize the mouse monoclonal antibody to make it a human. Essentially, almost an entire sequence of the antibody is humanized. So it's a humanized monoclonal antibody, and then you have to purify it, and you have to make it, and you have to make it in a facility that you can go into a clinical trial in humans. And so that's passive immunotherapy. You're actually come in and you inject the antibody containing solution IV, and you have to come back, and in Elan's clinical trial, they came back every 13 weeks and got another bolus of the antibody. It's expensive, and it requires repeated administration of the vaccine. One of the advantages of this is if you begin to develop adverse events, then you can eliminate the antibody by not delivering antibody. So the body will clear the antibody over time. Whereas if you actively immunize someone and you have an adverse event, you're not going to be able to turn the immune response off unless you start using immunosuppressive technology and techniques, and we've seen that can be problematic as well. So I'm just going to briefly talk about this says Pfizer now, and it's Janssen. Originally it was Elan, and so they've sold their immunotherapy to Pfizer, and then I think it was spun off to Janssen, a subsidiary or whatever. The bottom line is this: phase 1 and phase 2 clinical trials went pretty well. There were some adverse events associated in particular with people that were apolipoprotein E4 positive. They developed what was called vasogenic edema, and we actually at UCI, we had a subject that was in the clinical trial that did develop this vasogenic edema, and the patient died, and we worked up the neuropathology on that particular case. The bottom line is they split the trial up into people that were non-ApoE4 carriers and ApoE4 carriers. This refers to the study in which there was ApoE4. So in individuals, because there was higher levels of adverse events in the people that had ApoE4, they split the trial and they gave them lower doses of their bapineuzumab antibody. And so rather than spend a lot of time on this, I think we've learned things from this clinical trial. The endpoints weren't successful or positive. There was some evidence in the phase 2. There might have been a mild benefit to patients that in the trial that didn't have an ApoE4 allele. And I'll stop there. Yes. And then the other large clinical trial with the passive immunotherapy was by Eli Lilly, solanezumab. And that, interestingly, the antibody was targeting a different portion of the A-beta molecule. This also went through a phase 3, and safety was good, but there was, for the most part, no positive clinical outcome. So there's also a disappointment. And the last immunotherapy clinical trial, and I in my very first slide, I told you that immunotherapy, at least from my perspective, was going to be vaccine. This is another form of immunotherapy, and maybe you could consider it a passive immunotherapy. This is a therapy, IVIG or GammaGuard by Baxter. It's used in some types of autoimmunity, and you may have heard of patients receiving this. It's purified antibody molecules or gamma globulin, and it's given, and it's not clear exactly what the mechanisms are, but they spent a lot of time, and the phase 2 results were encouraging. But as almost every clinical trial in Alzheimer's, phase 2 always seems to look promising, and I've told people if I had something in phase 2 and it was promising, I'd sell it. Because what happens when you get to phase 3 seems to be there's no effect, and the drug, drug development is stopped. And I'm making fun, but it turns out, yeah, it's true. So I see some people shaking their heads. I put this as the next last slide, going back to what Adam was talking about earlier in the concept of when pathology occurs, A-beta, the tau, brain structure, memory, and then clinical function. So this is a long process over 15 to 25 years from the beginning of amyloid accumulation to real dementia in these individuals. And so my laboratory and many other laboratories, and you've heard other people say that we're probably going to have to intervene much earlier in the disease process, especially for an anti-A-beta or anti-amyloid therapy. We're going to have to go in earlier if we expect to see a clinical benefit. And that's, I'm a consultant for the DIAN, Dominantly Inherited Alzheimer's Network. And they're going forward. At least one of the therapies is going to be immunotherapy, and we're still deciding on what the other drug candidate or candidates are going to be, and we'll be meeting in a month and a half in San Diego at a meeting to discuss this as to what's going forward. The advantage here, especially when you've heard earlier people talk about these biomarkers are getting better and better, imaging and biochemical markers on in the cerebral spinal fluid. And so the hope is with these individuals that have these dominant mutations that we'll be able to put the therapy on before they have any clinical signs, and we'll be able to see some change in the imaging markers and some change in the CSF biochemical markers to give us an indication of whether these therapies are actually working in human patients. And with that, I'd like to wrap up, like everyone else, by acknowledging the people in my lab at UCI Mine. I have a major collaborator, Michael Agajanian. He's the director of the Institute for Molecular Medicine in Huntington Beach. And while we're no longer working with ICOR, I'd like to thank them for partnering with us to use their electroporation system. And like other people, I'd like to acknowledge both the federal grants from the National Institute of Health, as well as grants from, 2 grants from the Alzheimer's Association. And with that, I'll stop. Okay.
When you get your DNA vaccine, how will you get research participants? Are you going to get people with MCI? Are you going to get people at high risk for Alzheimer's? How will you select those?
So that's So let me back up just 1 second and say that many of the clinical trials that have been tried in the past, the patients didn't come through necessarily an Alzheimer's disease research center. And I think if you heard earlier in the day, the ability of the clinician, the neurologist, and the person doing the neuropsychiatric testing, those tests to determine what your cognitive ability is, maybe 15% of the people don't have Alzheimer's disease, and there's some indication that it may be higher than that. And that's one problem with clinical trials is, and I think with the imaging biomarkers and with the CSF biomarkers, we're going to get better at picking those people that we want in a clinical trial. In that Elan's vaccine clinical trial, of the 10 brains that came to autopsy and that were examined by a neuropathologist, James Nichol, only 9 of them had Alzheimer's disease. 1 of them had some other neurodegenerative disease. So when you start having people that don't have the disease and are in your clinical trial, that means you're going to have more people. And how long are you going to follow people before you see a positive clinical outcome? If you start with people that are moderately or severely demented, you've already seen the talk on the imaging. There's already enlarged ventricles, there's cortical thinning, there's synapse loss, there's neuronal loss. You're asking a lot. You're probably going to have to rely on Matthew Burton-Jones or somebody that's going to come in and provide nursing ability to try and nurture the brain. So we're not adding anything that's going to induce growth. We're just trying to get rid of the pathological features of the disease. And I probably didn't answer your question, did I? No. Can you repeat it? My wife accuses me of this all the time. So. Right. Well, we would go out and so like through our Alzheimer's Disease Research Center at UCI, we would partner, and for our DNA vaccine, we partnered with Lon Snyder at USC. And so we would need to first get approval, and then we would go out and through various Alzheimer's disease research centers and try and recruit people that would meet the criteria that based on what got approved by the FDA. Well, what we would like to do is immunize people that before they have clinical symptoms, but based on MRI and biochemical biomarkers of Alzheimer's disease, we'd like them to have some aspects of the disease. And we'd like to, so we'd be able to see, hopefully, some effect on those things by imaging and by the CSF. So we'd like to see an increase in A-beta, a decrease in tau in the CSF, and we would like to see the brain stop shrinking and the amyloid load go down. So you got tired of waiting? Did I answer it?
You answered it.
Oh, okay. So we would, like the Diane study, I think we would like to start, I've had discussions with my collaborator, Mike Agajanian, and neither one of us are MDs, so you need to be, you may not want to even... Well, for us, because it's a vaccine and it's an active vaccine, we would like to immunize people in their early 50s. I mean, so having said that, we've got to be confident. We've already seen with the Lanz clinical trial that we had serious adverse events. With the passive immunotherapy, there was vasogenic edema, and when they look, those people, most of them resolved, the vasogenic edema resolved, but when they looked 3 to 6 months later, in those areas where there was vasogenic edema, there was evidence by MRI of microbleeds. So it's not benign, and so we need to be very careful when we move forward, and that's the problem with, I said, as an active vaccine, you can't turn it. You can't just turn it off. You can quit giving the vaccine, and the antibody response is going to taper off over time. But that's. So I would like to go in very early and have a clinical trial over a long period of time. The problem with that is it's very expensive, and you have to find the money. The typical, I think, the clinical trials went from 12 months to 18 months, And if you talk to Lon Schneider at USC about clinical trials, one of the problems is the people that are not demented, your control group, sometimes they go down, sometimes they go up. So you may see something changing in the people that you immunize, but when your control group changes or gets worse, then you have no clinical effect. So these are problems. In mice, like I said, we don't have any problem curing Alzheimer's disease. It's the humans that represent the problem. So, any other? Okay, thank you.